All right, so I'm going to introduce you one more time. I already have the honor to introduce you this morning, and then this is going to be the second time for the technical seminar. So we're very happy to have Joan Rose with us today. She holds the Homer Nolan Chair in Water Research at Michigan State University, a member of the National Academy of Engineering since 2011, and the winner of the 2016 Stockholm Water Prize, an international expert in water microbiology, water quality, and public health safety. Recently, she's done a lot of work in collaboration with the state of Michigan on the statewide program on wastewater surveillance for COVID-19. And she's also in charge of the Global Water Pathogens project that she talked about this morning. She earned a PhD in microbiology from the University of Arizona, Tucson, and a master's degree from the University of Wyoming. We're very, very happy to have you here with us, Joan, and I'll leave the floor to you. Thank you.
Well, thanks so much. It's really a pleasure to be here. I just can't tell you after a year and a half of seeing everyone on Zoom, how great it is to be here and walk around the campus and see the students and see everyone and talk to everyone in person. So I so appreciate the invite. And I love talking about water microbiology. And just when I thought things would calm down, you know, a couple of years ago, right? Year and a half ago, COVID hit the scene. So I'm going to tell you a little bit about the environmental virology work that I've gone on, and then I'm going to end by talking about some of the SARS wastewater surveillance work that's going on, not only in Michigan, but around the world. So I'll just start at the beginning. I know we have a broad audience here. But of course, viruses are biological nanoparticles. They're quite small. They're obligate parasites. They need a host to replicate. They're generally considered very host specific when we think about viruses. But of course, we know about these jumps, you know, that viruses can make from one species to another. They're simple structures. They are RNA or DNA, surrounded by a protein coat. And of course, some contain lipids and glycoproteins. And our most famous virus that is now on the news quite a bit, the SARS, is one of those that has that envelope. So viruses infect all living entities. And we found now that, in fact, early on, I don't think we knew much about viruses of fungi, viruses of algae. We knew about the bacteriophage, right? The viruses that infect bacteria. We studied the human and animal viruses that cause disease, but there was a lot of viruses that we really didn't know very much about. And I thought it was really funny, interesting when we think about the traditional viruses, you know, that I work on, which are the enterics, the fecal oral viruses. When you start looking at these other kind of viruses, this alga virus in water actually has affinity for cells in the brain. And they found it in mice and humans. In the UK, they were calling it the stupid virus. And it was impacting cognitive functions. So some of our ideas about viruses are just really emerging, even though we've known about them for quite some time. I also thought it was interesting that, you know, viruses are so small. So when you think of the total amount of DNA, right, on Earth, they're just a small percentage of that DNA and RNA. But when you think of the entities, like a virus itself, they're actually quite abundant. They're considered the most abundant entities on Earth. And I just put this together. This is what we think, well, this is our estimate of humans and livestock. This is what we estimate, although it's a very rough estimate. We don't even have that in terms of biodiversity, the total number of individual animals. These are the bacteria, and that's where they think the viruses are. So, and most are non-culturable. So the characterization of the virus world is quite poor, actually. in terms of what we know about these entities. Now, when I think about the history, and there's key events, I think that really emerged out of new technology for virus discovery and study and characterization, the science around the viruses and virology, but health. So these things intersected really to promote what we know about viruses. Of course, it started with some of the clinical descriptions of diseases like polio virus. Yellow fever was one of the first diseases that, they knew it was a virus. We have, of course, the infectious filterable agents was identified. Vaccinia in 1913 was actually cultured. And so for culturing viruses, we need to know the host. And so we had to get cells, or we used animals. Generally, we used animals in the early days. I'm not that old, I might look old. But, and we still wanted to think about culture was a very important issue in terms of virus, the study of viruses. Bacteriophage, 1915, and of course, culturing the viruses out with bacteria became not only a way to study viruses, but a way to understand DNA, a way to look at life in general. So it became a very important tool in biology. By 1930s, they were culturing yellow fever and smallpox. Polio virus was isolated in 1950 from cell culture. And of course, we didn't actually see a virus until the 50s. We had no idea actually what it looked like until electron microscope was invented. By 1985, another key thing was PCR. So polymerase chain reaction allowed for the discovery or the characterization of RNA or DNA, within this world that we couldn't culture. And then the whole idea of metagenomics was first described in 1988. That is the idea of taking a sample and looking for all the genome in a single sample with new technology, new instrumentation that will give you the sequencing, right? And also the new bioinformatics that would let us link things up and discover things, right? How do we match up what the sequences mean? And probably metagenomics didn't really get going until the 1990s in terms of environmental use. Now, if we look at the history of environmental health in the world I live in, it was really well before we knew what viruses were. The diseases likely caused by viruses were described. And they were described in various, types of writings. Of course, epidemic jaundice, where it spread. They didn't actually know it was contagious, but they knew it spread. It's been described. And interestingly enough, historical records by armies are where we find a lot of information about disease, because they kept track of the soldiers and the diseases they got. And so they had a lot of epidemic jaundice recognized in these military troops through the Civil War and First World War. Of course, that was hepatitis. The disease was named infectious hepatitis. And by 1973, the virus was finally detected under the electron microscope. But really, from that time that they actually named it, it took 5 decades. And I just want you to think about this, because think what we just did with SARS and all the tools and techniques we have, and the emergence, the global emergence of scientists, immunologists working on this virus, how fast we characterized it, how fast we got vaccine out. This took 5 decades of studies on immunology until they developed a vaccine for hepatitis A. And we still have, despite that, even in the United States, we still have a lot of hepatitis A throughout the world, even though we have a vaccine. But hepatitis A and other enteric viruses could be looked for in wastewater. So we knew we could look for them in wastewater and study infections that came from the community in wastewater. But the main goal was really focused on sort of the sustainable development goals, which have been articulated right at the global level. And that is, how do we control these waterborne pathogens? And that means controlling feces, treating wastewater, so that we destroy the infectivity of the virus before we release it to the waterways and prevent waterborne disease. So the whole focus was, oh, these viruses are going to be there, but how much is there, and can we get rid of them through wastewater treatment? Recognizing that our indicator system may not have been doing well. But it's interesting, even with that, and even having vaccines and all this technology. So in 2017, I don't know if you remember, but we had a huge outbreak starting in California in the homeless of hepatitis A. So they were not vaccinated. They didn't have good hygiene. And it spread in that community and to others. And it spread across the United States. Probably about 30 states had outbreaks of hepatitis A. Michigan had an outbreak. Irene Zarwacki, and I know some of you know her, was monitoring Detroit, where there's homeless. And we had a huge outbreak in Michigan in particular, where we had, infections and death. And they were monitoring this in the wastewater as well. This whole idea of the infection spreading in the wastewater back then. Now, metagenomics opened things up even more, because if you think about PCR, if you know about polymerase chain reaction, I always kind of think of it as a copy machine, right? You have to kind of know what you're copying. So PCR, we have to design primer sets or little pieces of DNA to copy the genes that we want. So we have to kind of know what we're looking for when we're going to use PCR. But with metagenomics, when that came along, it really broadened the idea of who was there. Looking at different sources, looking at the community, and then doing sequence analysis to say who was there. Now, it wasn't quantifiable, and it didn't tell you they were alive. We still use culture for that. But it really opened our eyes. I wanted to talk real briefly about one of the studies that was done by a student from Korea in looking at viruses in ballast water. And I think the thing that happened with the metagenomics for the viruses is that in the past, they had looked at all the DNA in a sample. And so the bacteria, the eukaryotes, the fungi overwhelmed any virus that was there. The only viruses they actually found were some of the DNA phage that are there in such high abundance you could find them. So they didn't see any of the RNA viruses or any of the single-stranded viruses. And it was just a few families. So what we did is the world of studying viromes then started combining the traditional kind of culture techniques that we had used of concentrating water, separating out the virus particles, purifying them from bacterial contamination, instead of putting them on cell culture, putting them through the sequencing. So that when you got deep sequencing, you were really just looking at some of the viromes, doing reverse transcription, so you could get both RNA viruses and DNA viruses. So this whole methodology kind of evolved, where we could start to look at different kinds of waters and the whole virus community. I'm showing you this because I think we've underestimated some of these diseases that may be happening in the wild, to plants, to animals, and things like this. Because this really opened my eyes, I have to tell you. So we were in two ports, California, Long Beach, big port, and Singapore. We looked at harbor water, and then we also took ballast water from the ships that had picked up. They have to record. Everybody know what ballast water is? The water in the ship that keeps it from tipping over. So when you have cargo on, you have to release, or when you go into port, you can release the ballast water, or you release it at certain times. And so they knew ballast water was carrying invasive species around the world, but viruses had not really been studied. And so it was quite a challenge. Large volumes are concentrated for virus work, anywhere from 10 liters to 20 liters through ultrafilter. Sampling ballast waters, even though it was starting to be recommended by the International Global Consortium for ports, what they didn't really have any way to get the sample. You kind of dipped down into the ballast to get all these samples and then lowered them over the edge. Here's the Long Beach port, and people helping us in Singapore get this water. It's a lot of water that comes out of these ballasts. Now, I just want to show you a little bit of the result. First of all, richness, or how many different viral families are identified, the biodiversity, so to speak. And what we found is that, of course, most of the California, these are the green, Singapore's in the red. California was coming from the Pacific Ocean for the most part, fairly cold oceans. And so temperature was very, very different. That was one of the main things. And Singapore was coming, those ships were coming from the Indian Ocean and the Asian, area through there, and they had very warm waters. And we were trying to figure out how we got temperature of where the ballast water was picked up. Because once it got into port, the ballast water was the same temperature for everything, right? So we used lat longs of where they picked up the water and transposed that to average temperatures in those areas. And so we got average temperatures of where they were picking up the ballast. And in fact, then that was the main factor in terms of relationship with biodiversity, was that you were getting more, temperate, and more tropical waters from Singapore and more temperate temperatures from the California ballast waters. And, but I thought what was really interesting was, for me, the whole array of viruses that were found in the ballast waters. And a lot of these viruses, actually, I didn't know very much about. I had to look them up. I'm like, what is this virus? I thought the ranavirus is really interesting. So I don't know if you know about the ranavirus, but there's actually a group, a biodiversity group that you can join that's trying to save the frogs. The frogs are dying, and they were dying because of the ranavirus around the world. And so they're trying to save frogs. And so I found out that this is a virus that can jump from different species. And it's quite high mortality in the frogs. Doesn't appear to jump to humans as far as we know. So these were kind of the fish viruses we found. We found shrimp viruses, also shrimp viruses that could jump from freshwater shrimp to saltwater shrimp. If you looked in the literature, you found the same virus in these two kinds of species of shrimp, which was very not known. And we found some human viruses as well in there that I guess we weren't actually expecting. And when we looked at 3 different groups of viruses that we had found, the cyclovirus, the human cyclovirus is related to circoviruses. You may have heard of the circoviruses. This is a pretty new virus. But they impact pigs mostly. And what I found about the Cercoviridae family is that this family shows up in our wastewater. It's the smallest, one of the smallest viruses that we know of. So it's like 10 nanometers, way smaller than the polio virus, which is pretty small. And it's like, why is this pig virus showing up in sewage? And it's highly related to the human cyclovirus, which just recently emerged. It's a fecal oral virus. It actually killed off pigs in the United States about 2018, and pork prices went up. So I was studying this virus and said, what is this virus? I've never heard of it before. Why is it so abundant? So the cyclovirus, the B is ballast water, the D is disease, where disease has been found, and the H is harbor water. So this is where we found these sequences of these key viruses, a human virus, the Picnoviridae, the human cyclovirus, and the shrimp virus, where we could find that they were both causing disease here and there. They were found in the ballast waters, they were found in the harbor waters. And so, you started thinking about, disease and viruses in a broader perspective. So while we continue to struggle with the iconic virus risks, like such as polio and hepatitis, despite having vaccines, these new viruses are emerging and may be more important in key regions of the world than we know. Human, animal, and plant viruses are probably more important, and we have not studied those. So we don't really know how these are affecting some of the biohealth of the planet. And these viruses are much more able to jump species than we had traditionally thought about viruses being so host specific. Now, the enteric viruses that I work on cause a whole range of disease, both acute diseases like diarrhea, respiratory illness, and of course hepatitis, but they cause a whole slew of chronic diseases. Liver damage, kidney failure, heart disease, cancer, nervous system disorders, of course, ulcers, birth defects, and death are associated with these enteric viruses. Polio virus is probably one of those that is most well studied around the world. Of course, the disease symptoms were described early on. They called it infantile paralytic disease, even though it did affect some adults, but they were very worried because it affected infants, young people. They started recording epidemics by the 1900s. Contagion was identified by 1905. And by the 40s and 50s, they were culturing in primates and finally in cell culture, as I already mentioned. But actually, it wasn't until 1941, believe it or not, that they actually identified that this virus was growing in the intestinal tract. And that was its primary site of replication before it moved to the central nervous system and other parts of the body to cause disease. So by that time, by the 40s and 50s, people started saying, okay, let's look in sewage for this virus. Probably the broadest surveillance for a virus is polio virus. And it developed a global polio laboratory network run by the World Health Organization for polio. Now, this wasn't really, at the time they designed this program, it wasn't actually designed to do wastewater-based epidemiology. It wasn't designed to say, are we treating our wastewater appropriately and handling our fecal waste to meet SDGs? It was really looking at the vaccine, implementation and uptake of the vaccine. And so they developed and accredited laboratories in 92 countries across 6 regions of the world. And so this is a network of labs, an RNA virus that they'd already, for the last decade, right, couple of decades, been developing a program where they can monitor wastewater to look at how the disease is spreading, how well is the vaccine. I mean, the goal was eradication, right? And so smallpox was easy to eradicate compared to polio, which is still popping up here and there. And these are some of the previous cases that have been seen. And interestingly enough, there's also, since it's a live vaccine, there was some reversion to disease with some of the vaccine cases. So genetically, they were using PCR approaches and sequencing to distinguish wild type from vaccine-derived cases. Now, the vaccine-derived cases were a little bit, you know, they might have been milder in many of the cases. You can see that it's been very difficult to try to meet a goal over 20 some years of eradication. So wild polio virus has been eradicated in most continents, but then we have this vaccine strain. And so the disease is still considered endemic in key areas. And of course, early on, this is 2018, there was a lot of discussion about sewage surveillance in the fight against these type of diseases. So there's hundreds of diseases. If you're interested, you can go to the Global Water Pathogens Project. It's waterpathogens.org. And we do have a book that we redid that was done by the World Health Organization, the World Bank. And so we redid this book with a book online, a lot of chapters on the different types of pathogens. And it was really designed, it's got a global authorship, and it was designed to really focus on, can we treat sewage better before we discharge, to remove these groups of pathogens, whether it's the solid part, like the sludges, the solid parts that come out of a wastewater treatment plant, or the liquid parts. And I know some of you have gone to wastewater plants, maybe not everybody. So the mission for the Global Water Pathogens Project was really to be a knowledge resource and to guide the goals for sanitation and achieve safe water and technology. So that it built an online platform, 115 chapters, 7,000 scientific resources, et cetera, et cetera. And we have a network of 250 people that contributed from 52 countries. But you can join the GWPP, and right now we have about 800 membership who gets some of our news and that type of thing. Now, the reason I'm bringing this, I'll talk about this, but we're using this platform for developing tools to access the data, because this is very scientific. You can see that each chapter is designed to talk about disease burden, transmission, detection methods, occurrence in wastewater, persistent, and controls. And to really be quantifiable, to take, it's not necessarily a literature review, a complete literature review. It's taking the most significant papers that provided quantifiable data on the occurrence and concentrations of these viruses in wastewater. And so this is the virus chapters that we had. And this is what you get when you look at some of the data. It's really snapshot in time, a snapshot in time and location. So spatial and temporal snapshots around the world. This is, I think, the rotavirus and percent positives, percent negatives, and then they've got concentrations. So maybe 30 samples, 40 samples, 50 samples, right? That's what's in here. And the persistence information. So there were some tools developed to try to access the information for practitioners and that type of thing. So while the global international environmental virology crowd was working on these kinds of things, you know, and these viruses, lo and behold, a new virus emerges, a new pandemic type virus. Now, it wasn't that we didn't know about the coronavirus, we did. But of course, you probably know about the history of this. New outbreak of severe pneumonia by December 2019, probably some cases before that, but that's when it became noted. By January 2020, the scientists had isolated the novel coronavirus and designated it as SARS-CoV-2. By February 2020, the disease was named by WHO. By March 2020, WHO declared COVID-19 as a global pandemic. This is so fast. This is incredibly fast, just months, a few months, going from somebody saying we're having this outbreak in a location of the world to this global pandemic. Now, the family of viruses, they're positive single-stranded RNA virus, like polio virus, like the coronavirus. But of course, it has this envelope layer and glycoproteins on it, so it's not a naked protein. But this family of virus has been studied for quite a long time. First coronavirus discovered in 1932. And the viruses in this family affect a lot of animals all over the world. As far as I could find, 200 different hosts can support different coronaviruses. And of course, MERS and the first SARS, of course, the first SARS outbreak in 2003, that lasted 6 months, by the way. And then the MERS, which the largest part of the outbreak lasted 4 months, although MERS is still quite endemic, I think, in the Middle East. There's still, but it's not spread and seeing the same dimensions, but these bigger outbreaks lasted a very short amount of time compared to this new coronavirus we found. The other unique thing about SARS, and I can say that we have found coronaviruses in sewage. Some coronaviruses can infect the gut, replicate a little bit. So we might find them in wastewater. They're found in our genome studies, our metagenome studies. But nothing like this. This virus infects a lot of different cell types. And it was discovered early on that it could replicate in the upper intestinal tract. And it replicates to very high numbers in probably 40%. The early studies said 30% and 40 to 50% of the patients, this virus is replicating in the gut. So people were worried, is it waterborne? And they've shown prolonged excretion. So this is the numbers of virus that they found. in sputum, stool, throat, nasopharynx. And you can see up to 10 to the 8th per gram of this virus was found in stools. Now, this hit, the literature pretty fast. It was a scientist in China published that they found it in the intestinal tract and feces and also found it in wastewater. So I'm telling you, it was fast, very fast, that the environmental virology community took their methods for other viruses, took the primer sets and the techniques that had been used for PCR for these other viruses and adapted them for SARS, SARS-CoV-2. Now, there was a concern about waterborne disease. Could it be spread by water? It's found in water, could be found in high concentrations in water. But there were 2 lines of research, and I think there's going to be more that are coming out that have suggested not. And the first was simply a surveillance. They could not culture. They can culture the coronaviruses, right? We can culture it. We know how to culture these coronaviruses. And from infected patients, they could culture from the nasal passages, the throat, the sputum, but they couldn't culture from the feces. So there was 2 studies where they said they cultured it, but it could not be replicated by many labs who were trying to grow the virus out of feces. The second line of evidence, it said why. Why isn't we're not culturing? So when the virus is released from the upper intestinal tract, it goes through the colon, the lower intestinal tract. They believe that those spike proteins, those spiky things on the virus, are being inactivated by the colonic juices. So there's some inactivation, we believe, going on in the colon. It doesn't say that, there couldn't be live virus because sputum and other things go down the drain. If somebody's really sick and they do have diarrhea, and some of the patients did have diarrhea, I can tell you, it doesn't stay in the colon very long. You're right, it comes out fast. And so are we saying there's no chance that infectious virus is there? What we're saying is that we believe there's quite a large amount of inactivation. We don't know how much yet. But these are some of the studies that have led WHO and others to say that they don't believe this is a waterborne pathogen. So it's not considered a waterborne pathogen. Now, this created a paradigm shift for wastewater monitoring or wastewater surveillance. And you've now all heard probably about the wastewater-based epidemiology. But that terminology actually came from a group of scientists through the American Chemical Society and others globally that worked with them studying pharmaceuticals in sewage. They actually use that terminology. So the environmental virology group used surveillance. We talked about epidemiology, linking surveillance to epidemiology, monitoring, all of these kinds of things. But we didn't call our field wastewater-based epidemiology. They actually did. And by 2005, 2011, that terminology was used. And they were having conferences on it. And so what happened with SARS is that actually these fields have merged. So people that were doing drug surveillance started also doing virus surveillance. And they actually had pretty good experience going into the sewers, right? They worked with drug enforcement people, and they go from the wastewater plant into the sewer shed, to buildings, to communities, looking for the drug and the drug concentrations. And so they had that experience, whereas from an environmental virology, we were looking at what was coming in a wastewater plant, what was going out, could we treat it, right? What happens in a lagoon? We were looking at transport and fate. So this was a paradigm shift to really start thinking about, this is a way to survey what's happening in the community, not a way to look at waterborne disease. So there was a paradigm shift in our field. Now, I thought you guys might like to see the data. I took this 2 days ago. So the global data is about a month behind. This is August 8th. So new cases, 628,000. 7-day average, 607. These are just cases. United States, by September 14th, 150 159,000. Pennsylvania is at 3,732 cases by September 14th. Average is 4,000. And Michigan is at a lower, 200 cases, but average is 2,000 because we're on this upswing. Now, one of the things you'll notice between some of these and what happened in other states is we've had 3 waves in Michigan. We've had this kind of initial wave, which I think that we underestimated probably the cases and things. So who knows how accurate, right, the number of cases are. The second wave and this third wave that was really worrying people. We were trying to institute the vaccine and like, when is it going to come down? You know, you're here in April and you're saying, can it come down? Can it come down? It came down quite low, but we're starting to see this increase trend, which is why we've all gone back to masks, right? And even though we're vaccinated. So we had developed a network of laboratories across Michigan starting in 2015 for beach monitoring. And these were laboratories that are in public health, county health labs, in smaller universities, and some in utilities, but not very many. And they were all outfitted, got money through one of my colleagues, Dr. Shannon Briggs, who's amazing at the state, got money to equip our labs with PCR machines. So none of them had any kind of PCR machines in their biological labs. We got 15 labs established to do rapid monitoring on our beaches. Rapid monitoring for E. coli using PCR, rapid monitoring for microbial source tracking markers, human marker sewage, human sewage markers and cow markers, things like that. And of course, then the labs were positioned when SARS came along to take on a statewide program for monitoring SARS. Now, one of the key things that we learned about these networks is that you need training, there's rapid turnover, you need good quality supplies. And so all of these things we learned, we've got a couple of publications out about our network. But now we've emerged into a sewers program with some funding from the state for the next 2 years. We started with a pilot program. We now have 19 labs, 33 are local health departments, 57 counties, 6 Native American tribes, 18 labs, 19 tribes. And, we're looking at probably in a year about 87,000 samples around the state. So you can see we Upper Peninsula and the Lower Peninsula to do SARS monitoring. So quite interesting. One of the things we did in Michigan is try to develop a methodology that was robust, could give us precision, some accuracy, and ultimately we had to speed it up. Because one of the barriers for public health using our information was time to results. So we could do great stuff, get great data, but we would not get the results out to those who were making decisions fast enough. So it had to speed up. We were also going out into the sewer shed, which normally we don't, we go right to the wastewater plant. It's easier to get sample there. And we found that grab samples as you go closer and closer to buildings, you can imagine a grab sample, you might miss something, right? When did people flush? When did people shower? When do people do whatever they're going to do? And so developing a methodology for the state so that we could do composite sampling, rapid precipitation, it's called polyethylene glycol, a precipitation method, which is quite robust and forgiving for different kinds of samples that have a lot of stuff in it. And then we equipped all the labs with new instruments, and we moved to digital droplet PCR. So I don't know how many of you have heard of the digital platforms. There's about several companies now. QIAGEN is coming out with a new platform. But the difference between regular quantitative PCR, so we had PCR, I told you it was a copy machine, right? We moved from PCR to quantitative PCR. Quantitative PCR meant we had a standard curve and we followed the fluorescence as we replicated, right? 1 copy, 2 copies, 3 copies, you've got more fluorescence. We had to develop a standard curve to say what concentration was there. Well, what digital does is that it distributes the samples into tiny little drops, and it uses the most probable number. We don't need a standard curve. Because the sample is assayed in these little tiny drops, you have less interference with the PCR because we get interference with all the stuff in the sample. Even though we clean it up and extract it, we can get interferences with our PCR. So our laboratory network is using digital. And what you get is you get the negatives, you get the positives, you can use more than one fluorescence, so you can look for more than one marker. And as you train your machine, then you get a concentration of your marker in your sample. So let me show you some data really quickly. This is the first wave. As we started looking around the state, most of the first samples, which was pretty fast by April 2020, some of the first samples were being taken, April and May. And this was the first wave that we saw. Some pretty high peaks, but some communities were small. It didn't totally disappear, except in some communities, but we saw it go up and down. And we were also testing a variety of genes, like which gene targets should we look at? And ultimately, we arrived on 3 gene targets, and we left off the E gene, which is for the new project. This is the new sewers project. So the E gene in red, while all the genes were highly correlated, which is what we would expect, right? The E gene was less efficient in our instrumentation. We couldn't optimize it very well. So we're not using E gene anymore, using N1 and N2, 2 genes. And now we're looking for a 3rd gene, which gives you the variants, right? So our first wave, our second wave, and our second wave and our 3rd wave, and now this is where we are. And we came down, and we're coming back up in this 2021. So this is what one of the wastewater, and some of the patterns are very similar. Now, when we first started doing the campus, we were looking at the campus wastewater system. I don't know how it is here at Penn State, but our campus wastewater system goes to the East Lansing wastewater treatment plant. And the wastewater from our community runs through our infrastructure. So we could monitor East Lansing. We could isolate locations where we're monitoring the East Lansing wastewater and then monitor the wastewater coming from unique buildings. So we had to study the sewer shed and say, where are we getting? So this made some national news. I don't think anybody was too pleased about, this was early in the first wave. And of course, the bar had opened back up. And yeah, lots of social distancing here and masks, But what was interesting is that we found this outbreak. The outbreak was identified June 23rd. We found the outbreak on June 12th. We saw the peak in the sewage, but we didn't get it to public health fast enough. So when we went back to public health, we said, what's this peak? And they said, oh, we think this is the Harper's outbreak. So then that's when we streamlined things. We could see a slight peak when restaurants opened, Super Bowl was going on, things like that. But by March 15th, the following year is when we first started seeing the variants show up in the sewage. And it was just starting to show up in different places in Michigan. They had isolated it from cases, but in our county, they had only isolated it from one locale. But we were definitely seeing it in the wastewater signature. Now in the dorms, this is what happened. So this is a timeline. This is just 2021. I'm just showing you 2021 down to June. Moved in, immediately started a tick. Move that out of the way. Infection was seen in the different dorms, and you can see the green is the negative, but for the most part, we had positives throughout the semester. We had some dorms where we had sustained transmission and more cases showing up, and other dorms where it just popped in and then it went back down to low. So trying to figure out what went on in one dorm versus another in terms of could we do a better job of communicating and reducing the spread of some of this. Okay, let's see. I think I'm stuck here. I went backwards, didn't I? Okay, I'm going backwards. There we go. This is what happened over the summer. We had very few students in the dorms. We didn't monitor these dorms. We picked up these dorms in this fall semester. And so pretty much negative in the dorms where there's just a few students. They did get a peak here, but it wasn't sustained. We had some negatives. And we're starting now in September to start seeing the positives. We're seeing the positives all along in the East Lansing and the community show up. And this is what the variants look like. So this is the Alpha variant, which was known as the UK variant. So dates crossed here. And you can see when it started showing up, our suspect in February, we actually saw suspect positives and then confirmed positives by the time we got to March. It then went down to non-detects when the students were there, but we're seeing very low levels of that variant. But this is what the Delta variant looks like. We started looking at the Delta variant in July, and you can see that now that we've moved into September, we've got where there's positive COVID, it's Delta. And this, of course, matches what they saw. And again, we have some spatial issues with this is the community up here at the top, and these are all the dorms. So we could find it long before in the community, before we saw it in the dorms. That may have been a population issue, like there weren't enough people there, or it could be the ratio of number of infected to the total population. Well, finally, with this global phenomenon, we took the Global Water Pathogens Project, we have the book, we have the tools, and we got funding to start a COVID-19 platform. This is to take global data sets, and many of you may have seen Dr. Colleen Naughton's COVID Poops 19 site, and she is keeping an inventory through both literature, news items, press releases of who says they're monitoring for SARS in the world. Right now, there's 92 that have dashboards. There's 263 universities involved across the world in monitoring COVID, either for their community or for their campus. 57 countries and about 2,691 sites. This changes every day. I just got this a few days ago, but I bet it's changed by now. So we're using this to develop the wastewater sphere. We want to integrate data from across the globe. We want a central place where data can be saved. And, we didn't know how long this was going to last, but it's one place where we can have all the data that's going on in different places. Can we harmonize the data? People are reporting the data in different units, different ways they're normalizing the data. So we're trying to harmonize that so that that can all be captured. We're just going live now, so you can go take a look. Send us any notes if you see something crazy on there. But we still got some ways to go. Right now we have 14 dashboards, which represent 500 sites in 24 countries. We're working with, they're mostly high income countries, but we're working specifically with low and middle income countries that don't have their own dashboard, but are going to use this to deposit their data. So we're working with them to get their data up. We have country maps that give presence and absence, the most recent data, and you can do a toggle where you can look at what changed over the several months. And the data is all downloadable in CVS files. So we're going to have the data downloadable. And the data also, when you click on the individual spots, these are population based. So you can say, you know, what kind of population is being monitored. And it tells you what gene, what their average is, max, how many samples, and a little bit of information and more information about the site, because we have wastewater sites at the wastewater plant. We have sewer shed sites. We're also getting open canals. In Malawi, we're getting the river system because the river system receives their sewage. They're not really sewered. So we're getting river system there. And so we're going to be describing the metadata that goes along with this. We're trying to look at trends and statistical trends, taking the last 5 data. CDC is developing this too. We're going to try to see, and I think Europe is too, to try to harmonize it. We actually have information sheet that says how we're determining statistically from the last 5 samples, whether it's going down, whether it's going up, trending up slightly, or has not changed, or has no data, because some of these locations got sampled and then people stopped sampling them and added new sites. So it was trying to keep track of both the temporal and spatial data sets so that other people could take a look at it. Now, we do have a public health use case on the site where we're taking examples of how the public health programs are using the wastewater data for decision-making. So a leading indicator, early warning, and this is particularly when you have low prevalence of disease. Just looking at statistically whether they could, and this is true in Michigan, the data in Michigan, we did a survey of our public health labs, and they were using it to decide when they could go test. Use the wastewater data to go in and test nursing homes, other communities, that type of thing, to beef up the testing. Spatial risk maps, tracking of new variants, areas of concern, areas of populations, like I said, nursing homes, and of course, just communication and rapid the testing. Now we've got the vaccination. So this, what's going to be added to this is how do we use the wastewater information to look at vaccination? If our population is 90% and we still got SARS in the wastewater, are those breakthrough infections, or are those 10% the ones getting infected? What's happening in terms of how we implement the vaccination? And are those people getting sick? Overall, I'd just like to end with one thought, or several thoughts on where I think we need to look at research in general in the world of virology. And I really think we need to identify key natural and human environmental systems where we could characterize the viromes and try to understand more about plant, human, and animal health. We have to learn how to characterize the rare biome, the rare virus, because these are so small. And even though they're there in terms of abundance, they're still actually very important. How do we utilize new technology? And it's quite interesting to see that the instrumentation companies have suddenly come to Michigan and other places and said, would you evaluate this kit? Would you evaluate this new platform? Before they didn't, the environmental market is so small compared to the clinical market. It was like, we're begging them to come and help us modify instruments or collection methods or whatever, And now they, there is interest. They see that there's a need for this. And so I can see instrumentation and platforms and even collection systems changing the way we get the sample, we process the sample, we report the sample. And I think we need to understand not just who's there, but concentrations, and how viruses flow through various pathways. And we're still going to be quite interested in treatment technology. I mean, ultimately, we're sending SARS virus signal out of wastewater plants. SARS signal, PCR signal, can be found in effluents that are discharging to our rivers, our beaches, wherever it's going. We know nothing about the persistence of the signal. And ultimately, I think the wastewater industry is going to have to say, how do we get rid of that PCR signal? How do we get rid of a virus signal, whether it's live or dead? How do we get rid of an antibiotic resistance gene signal in our wastewater plant. These are the things that the wastewater plant's going to have to look at in the future. So I just want to acknowledge my group. They've all been working super hard. My colleagues really the global community it's been phenomenal to work with the global community on this through seminars and webinars and sharing information. And everybody's really willing to share their knowledge right now to combat this pandemic. And I really do think this is going to shift, the way we think about monitoring, environmental monitoring for the future. So thanks for having me here. It's really been a pleasure to give a real lecture standing up. It's been great fun. So I'll end there and see if there's any questions. Yeah.
Well, thanks for a great lecture. I wonder if there's names for all the numbers that we have, the exponents of some of those things. I mean, I know giga and tera and everything else, but it's some pretty big exponents there. I don't know if it's for the viruses in the world. But one of the things that, confounds knowing the spread of the virus is that if somebody gets sick, they don't report it, they go home, and then it never gets recorded. But you avoid that because you're measuring an action. So can you infer those numbers based on the wastewater to say, well, they're reporting this, it's really this.
Yeah. So there's a lot of modeling that's going on right now, and we haven't agreed on how we're going to do the modeling completely. So while I think the methodology for getting the signature is growing as a consensus, the interpretation is lagging behind, of course. And so the models are using a whole variety of different variables. One is like per person, how many grams of feces does somebody, on average deposit per day, right? Others are using fecal indicators to normalize the signal to say that that represents the fecal component. But with all these techniques, there's some uncertainty. And what we found in some recent publications, and we're looking at our data now the same way because they keep track of isolation and quarantines in each of the dorm. And so what they found was that new cases excreted a high load and then it came down and it was low excretion for a long time. So what you had is confounding. If you got a lot of people that got sick at the same time, you see this big peak spike. But then you see this long tail. If you have no new people, what if you have new people coming in and out? Then you have this confounding factor when you're trying to do that. The other thing is our leaky sewer systems. So if it rains, or also there's a lot of gray water being produced, you get a pollution signal. So yeah, yeah, I think E. coli or turbidity or any of these measures we may have to normalize. They're using some various indicators, but they show up in gray water too. So it's controversial right now. There's no consensus on how to normalize the data. And but I think we'll probably get there as we learn more. But certainly wastewater will get those that are asymptomatic that fail to report. And for most people, they have seen almost a 10-day lag between the wastewater signature and then being able to identify it as a case and did test it. I wouldn't say identify as a case because the symptoms show up here, you get the test, and 2 days later you get the result. So there's this lag between how we put the case in our column, but from a public health perspective, right? So it has its own uncertainties, as we already know. But we're getting correlations. I think initially public health was like, we're not going to use wastewater data. But in the Netherlands, they showed this very high correlation very quickly. The country adopted wastewater monitoring. CDC now has a program that's going to have a national surveillance program. And they funded different wastewater plants like in Philadelphia and other places to join the national surveillance. And our Michigan data is going to go up into the CDC dashboard. But there's, it's quite fascinating looking at all the global data sets and looking at them and what they've done, and even not knowing very much about the methods, just looking at the range of numbers, trying to translate it from what population was monitored, what conditions were monitored, whether it was a sewer, a river, a wastewater plant, and starting to get some hints as we look globally at what are the data going to tell us. So we're still got a long ways to go of getting there. But we're going to need modelers. We're going to need a lot more people that are looking at the uncertainty of the data, what assumptions can we make, and then how do we model
I'm
this?
sure the artificial intelligence community.
Yeah.
will come to your rescue.
That's right. Any other questions? Yeah.
I have a method question. I noticed in some of your data, you're looking at 3 genes. One would spike, but 2 would be 0. They would alternate. What is that?
Well, in some cases, it was very at the detection limit. And so when you're at the detection limit, you can get one that's spot just because of particles, and that's the most powerful number. So sometimes we're thinking also, before we had completely worked on the PEG method, that we weren't homogenizing the clumps. So that if you get a whole clump in one of your, your droplets, you get a different MPN. So we've got to have homogenization as they distribute the sample across all the either oil droplets to produce droplets, or their nano manufacture, the cells are manufactured with some of the new platforms. So we think some of that is that case, but the N1 and N2 gene generally, it's got about an 89% correlation. It's really high. And one is not necessarily higher than the other, but here they track each other very well. And with global data sets, everybody but one is either using N1 or N2 or both. And only one data set was using region early on, and they put monitoring. But everybody else is using N1 and N2. We've got new gene targets, which are the spike genes for the variants. So that's a whole different gene, which we'll get into. But the E gene was generally in our Michigan data set was generally below concentration wise on average than N1 and N2. And we had more negatives with the E gene than we did with the N1 and N2. So there was a sensitivity issue with that gene. Yeah, so we you'll see it like that every once in a while, but we analyzed in our pilot program, we had 40,000 samples from across the state. So we were able to analyze E gene and N1 gene. So we made a decision to drop the E and keep the N1 gene. So, but you'll see that every once in a while. Part of it is just, cosine distribution, but some of it may be the way our analysis. Yeah.
So you use the term sewer shed and have the opportunity on a campus with knowing where pipes are going to be the ones to monitor. Do you see that going to, say, a city scale, and people start, like the city of Detroit or the city of Philadelphia start trying to use their sewer shed, say, below hospitals or other sorts of things, or nursing homes, and target specific locations to monitor?
It's already happening in some communities. So some of our community sites are measuring correctional facilities. If you want to see a really interesting website that's monitoring correctional facilities, it's New Mexico. They actually name facilities. In Michigan, we keep them anonymous. So all our nursing homes and our correctional facilities are in our database, but they're not on the public site, so they're kept anonymous. But that's what they're doing. They're going directly to a building out in Victoria, in Australia, in Melbourne. They were monitoring hotels that were quarantine hotels. And until recently, that was working really well, but then saying that, okay, there's nobody else infected, or less somebody infected when they quarantine people coming into Australia. Recently, they had an outbreak, so the cases got out. And they're now monitoring other locations within the city, because before they only were monitoring quarantine hotels. And that was very, very well until just the last, I think, 6 months. where somehow the virus got out and they had an outbreak in an area they weren't monitoring that they didn't expect. And so then they started moving around in their sewer shack, looking at areas, sort of areas of population rather than building level. So we've got building level, we've got kind of areas, and then we've got the wastewater plant. Like the city of Philadelphia is doing the wastewater plants. So it's capturing different parts of the city from 3 different wastewater plants. Yeah, but you know, it's really difficult. I don't see us doing, I think what we need a lot of better understanding how to collect a sample. And I was looking at some of these robotics they have. They have sort of robotics that do things, and I was like, we need a little robot that goes in the sewer. It starts to get worse, and could go up the sewer and could start here and take a sample and then move up and take samples and things like that. But we're not, you know, we can place these samplers in some of our sewer sites, but some of them we can't. We have to be grass still. We can't actually get a sampler down into the manhole. So yeah, I think we, you know, I think we don't know enough about how to sample our sewer system. But they're learning now, and you can't isolate them. Yeah. Pretty cool. Pretty interesting.
So it looks it's very important and also helpful tools to identify the kind of conflict. So there are also require a lot of resources. What's your view about the pathway moved on from the rural area or the places with less resources?
Yeah, so we're doing some really small communities in Michigan, and we're doing some tribal areas. And so, you know, we had this one area that's a casino. So we're monitoring the casino and monitoring the community itself, and then at the wastewater plant. So 2 sites kind of upstream that got some populations. And I called the guy, said, you know, we have a kid, you know, and we were monitoring like on a Tuesday or something at the casino. And, you know, and it seems I'm like, you know, 2 people deposits while they're playing the slots. I don't know. You know, I wasn't thinking that maybe that would be very common. But they said later that, yeah, they had a case there. They had a case there, and then they had to do some more shutdown for a few days, clean up, enforce more mask wearing, and then reopen the casino. They didn't see an outbreak, but they did find a case, a couple of maybe 1 or 2 cases or something like that. So, you know, I think they're seeing that they can use it. Now, this is funded for 2 years, and it's not inexpensive. But the monitoring, the people monitoring is really expensive. And so I think they want to use it for saying, what's our detection rate? Like, can we monitor 100,000 people and see 1 case? Australia says they can find 1 case of 50,000. That's what they suggested. I don't know if that's true. So we've got to understand that. And then we've got to have an adaptive monitoring program where it's negative, 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 we decrease the frequency. And if it turns out positive, we start getting more samples or something like that, because I don't think it's sustainable to keep up the strategy that's in place right now for monitoring around the world, monitoring what's around the world.
Before we wrap up, this little gift from say to you for coming all the way and making the trip.
Oh,
There you
thank you
go.
so
Thank you.
much. Appreciate that.